Welcome to the deep dive, where we really trying to slice through all that complex information in your sources
Mhm.
and, unearth the most fascinating and, well, vital insights. Today, we're taking a truly essential deep dive
Really fundamental.
into something we all just do every few seconds about thinking. Breathing.
Yeah,
It's so easy to take for granted, right? But with every single breath, we're inhaling thousands of, well, microscopic hitchhikers.
exactly. And our respiratory system, it's the primary gateway,
The main door.
the main door for countless pathogens, which means infections here are not only incredibly common,
Right.
but also among the most damaging.
And there's this constant sort of invisible battle happening inside us.
Absolutely. It's truly remarkable. Our bodies have evolved these incredible layers of defense,
But the microbes?
but these microbes are so cunningly adaptable, always finding new ways past those defenses.
Wow.
So this deep dive, it's informed by the chapter Microbial Diseases of the Respiratory System
Yeah.
from Microbiology, an introduction, the 13th edition. And it's really going to illuminate this incredible biological warfare happening within us.
So our mission today, we want to unpack the major concepts, right? These microbial processes.
Understand the structures involved.
Get a glimpse of the lab techniques used for diagnosis.
Look at some of the most impactful diseases themselves.
And crucially, connect all of this to its real-world implications for you listening.
Yeah.
You'll basically get a quick but thorough understanding of this invisible world shaping our health with every inhale. So let's unpack this.
Let's do it.
Okay, let's start with the layout, the battleground itself. When we talk about the respiratory system, we generally split it in two, right?
That's right. The upper and lower systems. Makes it easier to conceptualize.
So the upper respiratory system, that's like the front porch.
Good analogy. It includes your nose, your pharynx, it's the throat, your larynx, or voice box, and all those connected areas like the middle ear, the sinuses, the tear ducts even.
And that connection is key, isn't it? You ever wonder why an earache sometimes follows a bad sore throat?
Exactly.
Mm.
It's because these areas are all linked up.
Mm.
Infections can and do spread pretty easily from one spot to another.
Okay, so that's the upper system. Then there's the lower respiratory system, the inner sanctum.
Precisely. This is deeper inside.
Oh.
It consists of the trachea, your windpipe,
Right.
the bronchial tubes branching off, and then those tiny, tiny air sacs called alveoli.
That's where the real action happens, the gas exchange.
That's where oxygen gets into your blood and carbon dioxide gets out. And just to give you a sense of scale,
Yeah.
your lungs have something like 300 million alveoli. It's astonishing.
300 million, wow.
Yeah, it gives you a gas exchange area that's, well, over 70 square meters.
Like a tennis court packed inside your chest.
Pretty much. It's incredible engineering.
Okay, so given how much air, packed with microbes, we're breathing in constantly, it's amazing we aren't sick all the time. How does the body defend itself?
Well, it starts right at the entrance. You've got physical barriers, coarse hairs in your nose. They act like a first filter. Then the mucous membranes lining your nose and throat. They moisten the air, but crucially, they trap dust and microbes in sticky mucus.
Right, the mucus trap.
But one of the real unsung heroes here is the ciliary escalator.
The ciliary escalator? What on earth is that?
Okay, imagine millions of tiny microscopic hairs, or cilia, lining your upper throat and down into the bronchial tubes.
Okay.
They are constantly beating, like tiny oars, all moving in one direction upwards.
So-
Yeah, creating this sort of conveyor belt. It moves that mucus with all the trapped particles and microbes up towards your mouth.
So you can swallow it or cough it out.
Exactly. It's a critical, continuous cleaning mechanism, constantly working.
That's amazing. And what if something gets past that? If it reaches the lungs?
Good question. If microbes do manage to get past the escalator and deep into the lungs, there are specialized immune cells waiting. They're called alveolar macrophages.
The macrophages, those are the eater cells, right?
That's right. They're phagocytic. They basically locate, gobble up, and destroy most invaders that make it that far. Plus, you have specific antibodies, IgA antibodies,
Right.
in your respiratory mucus, saliva, and even tears, providing extra immune protection. Layers upon layers of defense.
Now, here's where things get really interesting, I think. For, decades, scientists assumed healthy lungs were sterile, right? Nothing living down there?
That was the dogma, absolutely. A long-held belief. But recent genetic studies, things like the Human Microbiome Project, they've completely overturned that idea.
Blown it wide open.
Totally. We now know definitively that healthy lungs have their own microbiome, a community of microbes living there.
Seriously?
Yeah.
What kind of microbes? And how do they get there?
Well, the most common bacteria we find are genera like Prevotella, Veillonella, Streptococcus. And what's really surprising is their main source probably isn't the air we inhale.
No. Then where?
It seems most likely they migrate down from the mouth.
From the mouth. Wow, okay.
Yeah. And many are anaerobic. They don't use oxygen, which seems weird for the lungs, right?
Right. Very oxygen-rich environment.
But they've adapted. They produce antioxidant enzymes to survive down there. It's fascinating adaptation.
So, okay, lungs aren't sterile. They have a microbiome. What does this actually mean for us? What's the clinical relevance?
Well, connecting this to the bigger picture, it's proving to be a potential game changer. Researchers are seeing significant shifts in this lung microbiome in patients with chronic lung diseases.
Like asthma or COPD.
Exactly. Asthma, cystic fibrosis, COPD. They often find a different balance of bacteria, sometimes more gram-negative types.
Huh.
Understanding these imbalances, well, it could potentially lead to new treatments, maybe even probiotic therapies for lung diseases one day, like we're seeing for gut health.
Probiotics for your lungs. That's wild.
It is. And there's even some early evidence suggesting certain bacteria in asthmatic lungs might be linked to high nitric oxide levels, something doctors already measure. So it's a whole new frontier opening up.
Okay, fascinating stuff. Now let's shift gears a bit and talk about the specific infections, starting back in that upper respiratory system. A lot of common illnesses start here. We're talking pharyngitis, that's your basic sore throat. Laryngitis, which messes with your voice box, makes you hoarse.
Yep.
Tonsillitis, inflamed tonsils, and sinusitis, the sinus infection, which can give you a nasty headache if mucus gets blocked up.
All pretty common, unfortunately. The good news, as you mentioned, is most of these are usually self-limiting.
Meaning your body typically clears them up on its own?
Yeah, generally. But there are exceptions. There's one upper respiratory infection that's particularly threatening: epiglottitis.
Epiglottitis. That sounds serious.
It is. It's a very rapidly developing inflammation of the epiglottis.
That little flap that stops food going down the wrong pipe.
Exactly. And if it swells up, it can block the airway completely. It can be fatal in just a matter of hours.
Wow. What causes that?
Historically, it was often caused by a bacterium called Haemophilus influenza type B. But thankfully, the Hyba vaccine
The vaccine.
has dramatically reduced how often we see it now. Vaccination really works.
Okay, good to know. Speaking of bacteria, one we've all probably heard of, maybe even had strep throat.
Oh yeah, strep throat, caused by Streptococcus pyogenes, which is, interestingly, the same bug that can cause some skin infections like impetigo.
So what does it do in the throat?
It causes local inflammation, fever. It's quite good at evading our immune cells, our phagocytes.
Sneaky.
And it produces enzymes like streptokinases that break down blood clots and streptolysins that are toxic to our cells. These help it spread.
How do they diagnose it quickly? Used to take ages, right?
It did. Used to be overnight cultures. But now there are rapid antigen detection tests. You can get results pretty quickly.
That's helpful.
Very. Allows for prompt treatment, usually with penicillin, which is still effective. Though a negative rapid test might still need a culture just to be sure.
And sometimes strep throat leads to scarlet fever. How does that happen?
Right. Scarlet fever occurs if the specific Streptococcus pyogenes strain causing the infection produces a particular toxin, an erythrogenic or reddening toxin.
Okay.
And here's a cool microbiology fact. The bacterium only makes this toxin because it's been infected itself by a type of virus called a bacteriophage. The phage basically gives the bacterium the gene for the toxin.
Wow, bacteria get viruses too, and that changes the disease.
Absolutely. It leads to that characteristic pinkish red rash, high fever, and often a strawberry-like appearance of the tongue.
Sounds unpleasant.
It is, but usually it's fairly mild. The key is treating it with antibiotics to prevent a much more serious complication down the line called rheumatic fever.
Rheumatic fever, that affects the heart, right?
It can, yes. It's a serious inflammatory condition. So treating scarlet fever promptly is really important.
Okay, moving on to an even more serious bacterial infection, diphtheria, caused by Corynebacterium diphtheria.
Yes, and this is another one that's thankfully much rarer now in places with routine vaccination. It's part of the DTaP vaccine.
Why is it so dangerous?
Diphtheria is nasty because the bacteria can form this thick, tough, grayish membrane in the throat. It's made of fibrin, dead tissue bacterial cells, and it can physically block the airway. Suffocation is a real risk.
Terrifying.
And the bacteria itself doesn't actually invade deep into tissues, but similar to scarlet fever, if the Corynebacterium is infected by a specific phage,
It gets a toxin.
It produces an incredibly potent exotoxin. We're talking tiny amounts like 0.01 milligrams. Can be fatal.
Whoa. What's the toxin do?
It interferes with protein synthesis in our cells. It can spread through the bloodstream and damage the heart, the kidneys, even nerves, potentially causing paralysis.
So treatment needs to be fast.
Very fast. Antitoxin therapy has to be given really early before the toxin binds irreversibly to tissues. Prevention through vaccination is definitely the best strategy here.
Okay. Now, something maybe less terrifying, but very common and annoying, especially for parents. Otitis media, the common earache.
Yes, the earache.
Ah.
Often a complication of a cold or other upper respiratory infection.
What's actually happening in the ear?
Basically, pus and fluid build up behind the eardrum in the middle ear. This causes pressure and pain.
And why are kids more prone to it?
It's mainly anatomy. Children's auditory tubes, the tubes connecting the middle ear to the back of the throat, are smaller and more horizontal than in adults.
So they get blocked more easily.
Exactly.
Yeah.
Inflammation from a cold can easily block them, trapping fluid and allowing bacteria to grow. Common culprits are bacteria like Streptococcus pneumonia, Hemigen, and Haemophilus influenza.
Does the pneumococcal vaccine help with ear infections too?
Yes, it does. It's helped reduce the incidence of ear infections caused by the specific Streptococcus pneumonia strains covered by the vaccine. Another win for vaccination.
Okay, let's move to the viral side of the upper respiratory system and the undisputed champion, the common cold.
The most prevalent human disease.
Yeah.
Easily. And it's not just one virus.
Yeah.
Over 200 different viruses can cause what we call a cold.
200.
Yep. Lots of rhinoviruses, some coronaviruses, not the ones causing severe disease like COVID-19, but other common ones.
Why do we keep getting colds then if there are so many? Don't we build immunity?
We do. Gradually. You build immunity to the specific viruses you encounter. That's actually why older people tend to have fewer colds than children. They've encountered more of them over their lifetime.
Ah, okay. Symptoms are pretty familiar. Sneezing, runny nose, congestion.
Usually no fever, though. That's a key difference from the flu often. And these cold viruses, they tend to thrive at slightly lower temperatures.
Like in the nose and throat.
Exactly. The upper respiratory tract is a bit cooler than the lungs, which suits them.
How does it actually spread? Is it mostly coughing and sneezing?
Airborne droplets definitely play a role, especially in dry, cold air where they might stay suspended longer. But research suggests direct contact is also really important.
Like touching your face.
Touching contaminated surfaces, then touching your nose or eyes, that seems to be a major route of transmission.
Which brings up the age-old question. Why do colds seem to peak in colder weather? Is it just because we're all huddled indoors together?
That's certainly part of it. Closer indoor contact increases transmission opportunities.
But is there more to it?
There might be. Some theories suggest physiological changes in cold, dry air could play a role too. Maybe the cilia in our airways don't work as efficiently.
Ah, the ciliary escalator slows down.
Potentially.
Yeah.
Or maybe the nasal passages get constricted. It's likely a combination of factors, behavior, and physiology.
Right. Okay, so that covers the common upper stuff. But when these same microbes, bacteria, or viruses make it further down, that's when things can get more serious, right?
Exactly. When the infection moves down into the trachea, bronchi, or even the alveoli, we start talking about bronchitis, bronchiolitis, and the most severe complication, pneumonia.
Pneumonia involves the actual air sacs.
Yes, inflammation and often fluid filling the alveoli, which interferes with gas exchange. It can be caused by many different microbes.
Let's talk about some serious bacterial diseases of the lower respiratory system. Pertussis, whooping cough.
Right, pertussis, caused by Bordetella pertussis. It's a gram-negative bacterium with a particular target.
Which is?
It specifically attacks and destroys the ciliated cells lining the trachea, the windpipe.
So it takes out the ciliary escalator.
Precisely. It disables that crucial cleaning mechanism. Mucus accumulates, you can't clear it, and that leads to those severe, prolonged coughing fits characteristic of the disease.
It's mainly a childhood disease, right? I remember hearing about the whoop.
Yes, traditionally.
Yeah.
It has distinct stages. First, a catarrhal stage, like a common cold. Then the paroxysmal stage, with those violent, prolonged coughing fits that often end with that characteristic whoop sound as a person gasps for air.
That sounds awful.
It is. In small children, the coughing can be so violent it can even break ribs. And in infants, if oxygen levels drop too low during coughing fits, there's a risk of brain damage. Then there's a long convalescent stage that can last for months.
Is it treatable?
Diagnosis is often clinical, based on the symptoms, though lab tests like PCR are used. Antibiotics, like erythromycin, can help reduce transmission and might shorten the illness if given very early, before the paroxysmal stage really kicks in. But once those cilia are damaged, it takes time to recover.
So prevention is key.
Absolutely. The DTaP vaccine has been incredibly effective. We saw a huge drop in cases after it was introduced. But immunity can wane, which is why booster shots are important, especially for adults who might transmit it to vulnerable infants.
Okay, now for a really big one. Tuberculosis. TB.
TB, caused by Mycobacterium tuberculosis, a disease with a long, dark history. Centuries ago, it was a massive killer in Europe, maybe 20–30% of all deaths.
What makes this bacterium so tough?
It's a slow-growing, rod-shaped bacterium, but its defining feature is its unique cell wall. It contains large amounts of lipids, waxy substances.
It's like a protective coat.
Exactly. This makes it acid-fast in staining, meaning it resists decolorizing by acid alcohol. But more importantly, it makes the bacterium highly resistant to drying it, can survive for weeks in dried sputum, and also resistant to many disinfectants and antimicrobials.
A very resilient pathogen.
Extremely.
And you mentioned there are related mycobacteria?
Yes. There's Mycobacterium bovis, which causes TB in cattle bovine TB. It used to spread to humans via unpasteurized milk. Thankfully, pasteurization and testing cattle have made this very rare in places like the U.S. now.
Okay.
And there's also the Mycobacterium avium intracellular complex, or MA. These often cause infections in people with severely weakened immune systems, particularly late-stage HIV patients.
So how does Mycobacterium tuberculosis actually cause disease after you inhale it?
Well, for many people, their immune system actually handles the initial infection quite well. The bacteria might get inhaled, reach the lungs, and get ingested by those alveolar macrophages we talked about.
Right.
But Mycobacterium tuberculosis can actually survive and even multiply inside the macrophages.
Inside the immune cells? How?
It has ways to prevent the macrophage from destroying it. If the immune system can't eliminate them quickly, it tries to wall them off. It forms a lesion called a tubercle.
A tubercle, like a little nodule.
Yes, basically a collection of infected macrophages, other immune cells, and bacteria, surrounded by a fibrous layer. The idea is to contain the infection.
Does that always work?
Not always. If the disease progresses, the center of the tubercle can break down and liquefy, forming a sort of cheesy necrotic center. This is called caseous necrosis.
Okay.
This liquefied center actually becomes a perfect growth medium for the bacteria, and they can multiply rapidly inside this tuberculous cavity.
And if that ruptures?
If the cavity ruptures, the bacteria can spill out into the airways, leading to coughing up infectious bacteria, and they can also spread through the bloodstream or lymphatic system to other parts of the body: liver, kidney, brain, bone. That disseminated form is called miliary tuberculosis.
That sounds really bad.
It's very serious.
But lots of people seem to get infected without getting sick. Latent TB.
That's right. A huge portion of the world's population, maybe a quarter, is estimated to have latent TB infection. The bacteria are alive, contained within those tubercles, which might eventually calcify. These calcified lesions, called Ghon's complexes, can sometimes be seen on chest x-rays.
And people with latent TB aren't sick? They can't spread it?
Correct. They have no symptoms and are not infectious. But the bacteria are still there, and the infection can reactivate later in life, maybe if their immune system weakens.
And active TB, what are the signs?
Active TB involves symptoms: a persistent cough, often lasting weeks or months, sometimes producing sputum stained with blood, chest pain, fever, night sweats, and significant weight loss, which is why it used to be called consumption.
Diagnosis used to be slow, right? Looking for acid-fast bacilli under a microscope.
Yes, and microscopic examination can miss cases, especially if there aren't many bacteria. Culturing Mycobacterium tuberculosis is definitive, but it's very slow growing. It can take weeks.
Weeks? A long time to wait.
It is, which is why newer methods are so important. There are blood tests now, interferon gamma release assays, or IGRAs, that measure your immune response to TB antigens.
Faster.
Much faster results than culture, maybe a day or so. And there are nucleic acid amplification tests, NATs, that detect the bacteria's genetic material. These can give results in hours or a couple of days, and can even detect resistance to some key antibiotics like rifampin.
What about the skin test, the little bubble on the arm?
The tuberculin skin test, or Mantoux test. That's still a common screening tool. A small amount of purified protein derivative from TB bacteria is injected under the skin. If you've been previously infected or vaccinated with BCG, your immune system reacts, causing a raised, hardened bump after 48–72 hours.
So a positive test means active disease.
Not necessarily. That's crucial. A positive skin test just means you've been exposed or vaccinated at some point. It doesn't distinguish between latent infection and active disease. You need further tests, like a chest x-ray and sputum tests, to determine if it's active.
Okay. Now, treatment. Why is TB so notoriously difficult to treat?
Several reasons. First, as we said, the bacteria grow very slowly. Many antibiotics work best against rapidly dividing cells.
Ah.
Second, they can hide inside macrophages, which protects them from some drugs and immune responses. And third, resistance develops easily if you only use one drug.
So you need multiple drugs.
Absolutely. Standard treatment for drug-susceptible TB involves a combination of 4 first-line drugs, usually isoniazid, rifampin, parsenamide, and ethambutol, taken for at least 6 months. Adherence to that long regimen is critical, but can be challenging.
6 months. Yeah, that's tough.
It is. And the really big problem now is the emergence of drug-resistant TB.
MDR-TB.
Right. Multi-drug-resistant TB, MDR-TB, is resistant to the 2 most powerful first-line drugs, isoniazid and rifampin. Treatment requires second-line drugs, which are often less effective, more toxic, and need to be taken for much longer, up to 2 years.
And then there's XDR-TB.
Even worse. Extensively drug-resistant TB, XDR-TB, is resistant to isoniazid and rifampin, plus resistant to the best second-line drugs, fluoroquinolones, and at least one injectable agent. XDR-TB is incredibly difficult, sometimes impossible to treat, especially in patients who are also HIV positive.
That's terrifying. Are there new drugs?
There are some newer drugs, like bedaquiline, which offer hope, but resistance can develop to those too. It's a constant battle. Drug resistance is a major global health threat.
Is there a vaccine widely used?
There's the BCG vaccine, made from an attenuated strain of Mycobacterium bovis. It's widely used in many parts of the world, often given to infants. It seems to offer good protection against severe forms of TB in children, like TB meningitis.
But not used much in the U.S.
Less so. Its effectiveness against pulmonary TB in adults is variable, and it causes a positive reaction to the tuberculin skin test, which can complicate screening efforts in low-incidence countries like the U.S.
And TB is still a huge global problem.
Massive. It remains a global pandemic. Over 10 million people fall ill with TB each year, and nearly 2 million die from it, making it one of the top infectious killers worldwide. There's also a strong link with HIV. People with HIV are much more susceptible to developing active TB.
Wow. Okay, let's move from TB to bacterial pneumonias more generally. You said pneumonia isn't just one disease.
Right. Lots of different bacteria can cause pneumonia. We talk about typical pneumonia and atypical pneumonia, though the lines can blur.
What's the most common cause of typical bacterial pneumonia?
That would be streptococcus pneumonia, also called the pneumococcus. We already met it, causing ear infections.
Right.
It's a gram-positive coccus that often has a thick capsule around it.
And the capsule helps it.
Big time. The capsule makes it resistant to phagocytosis, harder for those macrophages to engulf it. It typically causes high fever, difficulty breathing, chest pain, and often produces rust-colored sputum.
How is it diagnosed quickly?
Besides clinical symptoms and x-rays, there's actually a very useful rapid test. It detects the pneumococcal capsule antigen in a urine sample. It can give results in about 15 minutes with pretty high accuracy.
15 minutes. That's great for starting the right treatment.
Absolutely. And again, we have pneumococcal vaccines that protect against the most common serotypes or strains of this bacterium.
Okay. What other bacteria cause pneumonia?
Haemophilus influenza can also cause pneumonia, especially in young children and older adults, or those with underlying lung disease. Again, the He vaccine has drastically reduced its incidence in kids.
Now, what about walking pneumonia? That sounds less severe.
It often is, though it can linger. Walking pneumonia is typically caused by mycoplasma pneumonia.
Mycoplasma. That's different, right?
Very different. Mycoplasmas are unique among bacteria because they don't have cell walls.
No cell walls. How does that affect things?
Well, it means antibiotics that target cell wall synthesis, like penicillin, won't work. It also standard methods. They often get confused with viral pneumonias initially.
What are the symptoms like?
Usually milder than typical pneumococcal pneumonia, low-grade fever, a persistent cough, headache. It often lasts for several weeks. Treatment is usually with antibiotics like tetracyclines.
Okay. Then there's legionellosis, or Legionnaire's disease. Famous name.
Yes, named after that 1976 outbreak at an American Legion convention in Philadelphia. It's caused by Legionella pneumophila.
And where does this one live?
It's an aquatic bacterium found in natural water sources like streams and lakes. But it becomes a problem when it gets into human-made water systems and aerosolizes.
Like air conditioning.
Exactly. Cooling towers for large air conditioning systems are a classic source. Also whirlpool spas, humidifiers, even decorative fountains. It can thrive in the biofilms within these systems.
How do you get it breathing it in?
Yes, by inhaling aerosols containing the bacteria. Importantly, it's generally not transmitted from person to person.
Interesting. And it's resistant to chlorine.
Surprisingly resistant, yes.
Yeah.
And it can even survive and replicate inside certain free-living amoebas found in water, which might help protect it.
Who's most at risk?
Men over 50, smokers, and people with chronic underlying diseases or weakened immune systems are at higher risk for severe disease. Diagnosis can involve culturing it on special media or detecting its antigens in urine.
Let's quickly cover a few others. Psittacosis. That sounds like birds.
It is also called ornithosis, caused by Chlamydophilus psittaci. It's associated primarily with psittacine birds, parrots, parakeets, cockatiels, but also other birds like pigeons and turkeys.
How does it spread?
Usually through inhaling dust from dried bird droppings containing the bacteria. The bacteria are obligate intracellular parasites, meaning they have to live inside host cells. Stress can make birds shed more bacteria, and stress can make humans more susceptible too. Symptoms are flu-like, fever, cough, headache, chills.
Q fever. What's the Q stand for?
Query, because its cause was initially unknown when it was first described in Australia.
Ah. What causes it now?
It's caused by Coxiella burnetii, another obligate intracellular bacterium. It's quite unique because it forms an endospore-like body that's very resistant to heat and drying.
How do people get Q fever?
Typically from infected animals, especially farm animals like cattle, sheep, and goats. Transmission can occur through unpasteurized milk, but more commonly through inhaling aerosols from contaminated environments like barns or pastures, where dried birthing fluids or feces are present. It's highly infectious. Potentially just inhaling a single organism can cause infection.
Wow. What are the symptoms?
A wide range. Many cases, maybe 60%, are actually asymptomatic. Acute Q fever usually involves high fever, severe headache, muscle aches, cough. A concerning feature is that it can sometimes lead to chronic Q fever months or years later, often presenting as endocarditis, inflammation of the heart lining.
Tricky to diagnose then if it shows up much later.
Very tricky. Treatment is usually doxycycline.
Okay, that's a lot of bacteria. Let's switch to key viral diseases of the lower respiratory system.
Right. Viruses are also major players down there. Viral pneumonia itself can occur, sometimes as a complication of other viral infections like influenza or measles.
And we've certainly seen serious viral pneumonias with newer viruses.
Absolutely. Coronaviruses like SARS-CoV, which caused the SARS outbreak in 2003, and MERS-CoV, which emerged in 2012, caused significant outbreaks of severe viral pneumonia. And of course, SARS-CoV-2 causing COVID-19.
What about RSV, respiratory syncytial virus? You hear about that a lot with babies.
Yes, RSV is incredibly important, especially in pediatrics. It's the most common cause of viral respiratory disease, like bronchiolitis and pneumonia, in infants and young children worldwide.
Is it serious?
It can be very serious, even life-threatening, especially for premature infants or those with underlying heart or lung conditions. Almost all children are infected by age 2, but it's not just kids. RSV is also a significant cause of serious respiratory illness and death in older adults.
Why syncytial virus?
Because it causes infected cells in the respiratory tract to fuse together, forming large, multinucleated cells called syncytia. That's a hallmark of RSV infection. Symptoms often include coughing and wheezing.
Do we get lasting immunity?
Unfortunately, natural immunity after infection is poor, and reinfections are common throughout life. There's a protective monoclonal antibody treatment, palivizumab, available for high-risk infants, and vaccine development is ongoing, with promising results for maternal vaccines to protect infants.
Okay, and now the big one we all dread each winter. Influenza. The flu.
The flu, characterized by those sudden onset symptoms, chills, fever, headache, muscle aches. Importantly, it's a respiratory illness, not the stomach flu that's usually caused by other viruses or bacteria.
What makes the influenza virus tick? You mentioned spikes.
Right. The influenza virus has an outer envelope studded with 2 key types of protein spikes. First, there are hemagglutinin spikes, or HA spikes. These help the virus attach to the host cells in our respiratory tract. They're also the main target for the antibodies our immune system produces after infection or vaccination.
Okay, HA for attachment. What's the other spike?
Neuraminidase spikes, or NA spikes. These have an enzymatic function. They help the newly formed virus particles detach from the infected cell surface so they can spread to infect new cells.
HA and NA. You often hear about flu strains like H1N1 or H3N2 that refers to these spikes.
Exactly. There are different subtypes of HA, like H1, H2, H3, and NA, like N1 and 2. The combination determines the strain designation, like H1N1 or H3N2, which were major players in human influenza for many years.
So this brings us back to the crucial question. Why do we need a new flu shot every single year? Why doesn't one shot give lasting protection?
That's the million-dollar question, and it comes down to the virus's remarkable ability to change. It does this in two main ways: antigenic drift and antigenic shift.
Drift and shift. Okay, what's drift?
Antigenic drift refers to small, gradual changes, basically port mutations that accumulate in the genes encoding the HA and NA spikes as the virus replicates.
Little typos.
Kind of. Over time, these small changes can alter the shape of the HA and NA proteins just enough so that the antibodies we developed from a previous infection or vaccination don't bind as effectively anymore.
So our immunity becomes less protective.
Exactly. The virus drifts away from our existing immunity. This is the main reason why the flu vaccine composition needs to be updated almost every year to try and match the strains that are predicted to circulate.
Okay, that's drift. What's shift? That sounds more dramatic.
It is. Antigenic shift involves major, abrupt changes in the HA or NA spikes, or both. This happens when different strains of influenza, a virus, infect the same host cells simultaneously, often in animals like pigs or birds.
A mixing vessel.
Precisely. Influenza viruses have segmented genomes. Their genetic material is in several separate RNA pieces. If two different strains infect the same cell, these RNA segments can get reassorted or mixed up when new virus particles are assembled.
Like shuffling a deck of cards.
Good analogy. You can end up with a brand new virus that has a completely novel combination of HA and NA spikes, maybe an avian HA spike combined with a human NA spike.
And our immune systems wouldn't recognize that at all.
Exactly. Because it's a major change, most people would have little or no pre-existing immunity to this new subtype. This is what can lead to widespread epidemics or even pandemics, because the virus can spread rapidly through a naive population.
Like the historical pandemics.
1918.
Yes, the devastating 1918 Spanish flu pandemic, an H1N1 virus, locally of avian origin, the 1957 Asian flu, H2N2, the 1968 Hong Kong flu, H3N2, and the 2009 H1N1 swine flu pandemic were all caused by antigenic shifts.
The 1918 pandemic was incredibly lethal, wasn't it? Killing millions, especially young adults.
It was horrific. Estimated 20 to 50 million deaths worldwide. And yes, unusually it had a very high mortality rate among healthy young adults between 20 and 40 years old.
Oh, them. The leading theory is that it triggered an overly aggressive immune response, a cytokine storm, in people with strong immune systems. The immune system basically overreacted, causing massive inflammation and fluid buildup in the lungs, leading to rapid respiratory failure and hemorrhaging.
Just overwhelming the body's own defenses.
Essentially, yes. The virus itself was virulent, invading the lungs deeply, but the host's immune response likely contributed significantly to the lethality.
How do we diagnose flu now and treat it?
There are rapid influenza diagnostic tests that can detect influenza A and B antigens from a throat or nasal swab, often giving results in about 20 minutes. More sensitive PCR tests are also used, especially for tracking circulating strains.
And treatment.
There are antiviral drugs like oseltamivir, Tamiflu, and zanamivir, Relenza, which are neuraminidase inhibitors. They target the NA spikes. If it started early, within the first day or two of symptoms, they can reduce the severity and duration of illness. And of course, antibiotics might be needed if a secondary bacterial pneumonia develops, which is a common complication.
And vaccines. Are we getting closer to a universal one?
That's the ultimate goal. Making a vaccine that gives long-lasting, broad protection against all strains is a major challenge because of drift and shift. Current vaccines are usually trivalent or quadrivalent, meaning they target 3 or 4 anticipated strains.
How are they made each year?
Traditionally, most flu vaccines have been produced by growing the virus in fertilized chicken eggs, a process that takes months. Newer methods include cell culture-based vaccines and recombinant vaccines using insect cells, which can be faster and are suitable for people with egg allergies. But the holy grail remains a universal flu vaccine, perhaps targeting parts of the virus that are more conserved and don't change as much as the HA and NA spikes. Research is ongoing.
Okay, one last category. Fungal diseases of the lower respiratory system.
Right. Fungi are all around us, and many produce airborne spores that we inhale constantly. Usually our immune system handles them fine.
But not always.
Not always, especially for people with weakened immune systems. We're actually seeing an increasing rate of serious fungal infections, partly due to factors like HIV/AIDS, immunosuppressive drugs used for transplants or cancer therapy, and broader use of antibiotics that can disrupt normal bacterial flora. These fungi are often opportunistic invaders.
So which fungi cause respiratory problem?
Several are important. One is histoplasmosis, caused by Histoplasma capsulatum. It's a dimorphic fungus, meaning it can exist as a mold in the environment and a yeast in the body.
Where do you find it?
It's endemic, meaning commonly found in soil in certain regions, particularly around the Mississippi and Ohio river valleys in the U.S. It especially likes soil enriched with bird or bat droppings.
Bats carry it.
Bats can carry it, yes. And bird droppings provide nutrients for the mold to grow. People usually get infected by inhaling airborne spores called conidia, especially when soil is disturbed, like during construction or cleaning old chicken coops.
What's the disease like?
Often, it's asymptomatic or causes mild flu-like symptoms. But in some people, especially those with weakened immunity, it can cause a more severe illness resembling tuberculosis, or it can disseminate to other organs.
Okay. What about coccidioidomycosis? Valley fever.
Another important dimorphic fungus, Coccidioides immitis, or C. posidacea. This one is found in the dry, alkaline desert soils of the southwestern U.S., parts of Mexico, and Central and South America.
Valley fever, I've heard of that, especially in California and Arizona.
Exactly. It's transmitted by inhaling airborne arthroconidia, which are barrel-shaped spores released when the soil is disturbed by wind, construction, farming, or even earthquakes. Just driving through an endemic area during a dust storm can potentially lead to infection.
Is it usually serious?
Like histoplasmosis, most infections are asymptomatic or cause mild, self-limiting flu-like or pneumonia-like symptoms, but it can progress to severe pulmonary disease or disseminate, particularly in certain ethnic groups like Filipinos and African Americans, and immunocompromised individuals.
Then there's pneumocystis pneumonia, PCP. That has an interesting history, right? Was it a protozoan?
It does. For a long time, Pneumocystis jirovecii, it used to be called P. carini, was classified as a protozoan. But genetic analysis eventually showed it's actually a fungus, albeit a very unusual one.
And it causes disease mainly in...
Primarily in severely immunosuppressed individuals. It was one of the major opportunistic infections and a leading cause of death in people with AIDS before effective HIV therapies became available. It's actually thought that many healthy people carry it in their lungs without any problems, but it causes severe pneumonia when the immune system is compromised. Without treatment, it's almost always fatal in those cases.
Wow. Are there others?
Yes, several others. Blastomycosis, caused by Blastomyces dermatitis, is another dimorphic fungus endemic to parts of North America, particularly around the Great Lakes and Mississippi River Valley. It can cause pneumonia-like illness and skin lesions. And then there are ubiquitous molds like Aspergillus, found in soil and compost, and Rhizopus or Mucor, which can cause devastating infections, aspergillosis and mucormycosis, mainly in highly immunocompromised hosts, like transplant recipients or patients with leukemia.
So these opportunistic fungi really highlight the importance of a healthy immune system.
Absolutely. They underscore how critical our immune defenses are in constantly keeping these environmental microbes in check.
Phew, that was a truly deep dive, covering a lot of ground in our respiratory system. From that amazing ciliary escalator constantly cleaning,
Do its job.
to the incredible adaptability of viruses like influenza with its drifts and shifts, and the sheer resilience of bacteria like Mycobacterium tuberculosis. It really makes you realize our body is this complex, active battlefield.
It really is.
Yeah.
We've seen how microorganisms constantly challenge our defenses, from those simple physical barriers right up to sophisticated immune cells and antibodies. And importantly, how our scientific understanding and our diagnostic tools are constantly evolving to try and keep pace with these threats.
Yeah, identifying things faster, developing better treatments.
Exactly.
So you listening, you've learned about these silent, invisible battles happening with every single breath you take, about the surprising discovery of the lung microbiome, and the intricate ways serious diseases like diphtheria, pertussis, TB, and the flu leave their mark. It gives you a new appreciation for breathing, maybe.
Definitely. And think about that constant evolutionary dance, particularly with influenza's antigenic changes and the terrifying rise of drug resistance in bacteria like TB. It really poses a critical question for the future, doesn't it?
Which,
as these microbes continue to adapt, evolve, and sometimes defy our best treatments, what new strategies, what innovations in public health, in microbiology, in vaccine development will be absolutely essential for protecting ourselves, protecting humanity in the coming decades?
that is certainly a big thought to mull over. Thank you so much for joining us on the deep dive. We hope this exploration of the microbial diseases of the respiratory system has given you a fresh, maybe deeper perspective on this vital area of human health. Until next time, keep breathing deep.
And stay curious.